Good morning.
Good morning. Okay, we've got just over 3 minutes left to knock out the warm-up. It's a pretty short and easy one, and I do want to go over it. So go ahead and knock that out for me, and then we can talk about it.
Good morning.
Good morning. So really quickly, hopefully you had an opportunity to knock out the warm-up. I want to go over it because I think there's some important stuff here, and it's just a good habit to be in. Let me see. All right. So question 1 asks, a cell spends most of its life in which phase? Can someone help me out with that?
Interphase.
Interphase, good.
Interphase.
Excellent, thank you. Interphase is the longest phase of the cell cycle. So that's where the cell is for most of its life. Okay, now we're trying to match the organic compound with its subunit. So what's the subunit of a protein?
Amino acid.
Excellent. What about a lipid?
Glycerol and 3 fatty tails.
Good. What about carbs?
Glucose.
Excellent. And then nucleic acids?
Nucleotide.
Good. So we've got a lot of information here. This is unit 1 stuff, but it's still going to be relevant really throughout the course. And of course, this course is cumulative. The EOC exam in January will cover everything that we've learned from, August 23rd or 24th until now, or I'm sorry, until January. So it's a lot that we need to be able to remember. That's why we continue to touch on these things. So it's not just to make you all feel bad. It's just this practice.
Mr. Red?
Yes.
You said we're taking the test on the EOC in January?
That's currently the plan.
Because I was told we were taking all EOCs in October, and our teacher even had like times and schedules, and I'm like, but I'm confused because I got told like 3 different times: October, December, and January. But most of them have been October.
Okay, well, you've got to keep in mind that an EOC stands for end of course.
End of course.
Right.
Yes,
So
no.
we can only, we would only take the end of course exam at the end of the course. So
why did you just keep saying it's October then?
maybe that's
the quarter ones. Like for class that we have, like we're gonna take one for this class too. It's gonna be like a little small midterm.
Right. So we will have a midterm exam in October at the end of the first quarter. And the midterm exam is it's like a final in some sense, because it's worth a little bit more than just your average test. But it is not the same thing as the EOC. The EOC is a state administered exam. So the EOC, all students across the state of North Carolina who are in biology will take that exam. And that's given at the end of the semester in January. Okay, hopefully that answers the question. Okay, so this one says, true or false, during the process of DNA replication, 2 identical strands of DNA are created.
True.
Yeah, now there's some caveat. There is a caveat with that. When we say identical, they are copies of one another, but each of them is made up of a strand from. It'll be easier for me to explain this with a picture. Trying to choose a good one. So the new DNA, they are exact copies of one another, but if you can see, there's an orange color and there's a blue color. So the blue is from the original strand of the original double helix. The orange is then essentially attached to those original strands to create 2 new strands. So each new strand is made up of half of the original strand and half of the new strand. So in some ways, they are not exactly identical because each original strand is slightly different. And of course, there are potential mutations, but they are copies of one another. So that's kind of what we mean by identical here. Okay, so what's the first step of the cell cycle? This is what we talked
The
about
first
yesterday.
one is G1.
Good. The first one is G1. Then what
And
about
then
the
the
second
next
one?
one is synthesis.
Excellent. In this case, we just have S. Then the third one is
G2.
good.
I think the fourth one is mitosis.
Excellent. And then the last one?
The C word. I just don't know how to pronounce it.
Yes, good. Cytokinesis. All right, so can someone just tell me quickly what happens in these stages? So in the first stage, Ayana told us that that's G1. What happens in G1? For me, I always like to think about G as standing for two things. One of them is gap, and then the other is growth. Yeah. So G1, the cell is growing. What about S? What happens in synthesis? Alicia, do you know? Does anyone know what happens during synthesis?
Something about the DNA.
Yep. Yep. So in synthesis, we're synthesizing new DNA. We're copying the DNA. We're making a second version of the DNA. So the timer is going to run out. But G2 is another stage of growth as the cell prepares itself to be split. Mitosis is what we'll talk about today. And mitosis is a somewhat complicated process, but essentially the cell copies itself. And then cytokinesis is when the cell moves. Cyto is cell, kinesis is movement. So the cell moves apart. It splits into two new cells. And then our last question, what is the purpose of checkpoints throughout the cell? Why do we have checkpoints?
To prevent any mistakes from occurring in the new cell.
Excellent. Thank you, Castro. Good. So we're going to talk more today about the specific process of mitosis. But before we do that, I do want to acknowledge that today is September 15th, and it is the start of National Hispanic Heritage Month. And National Hispanic Heritage Month is a celebration of all of the contributions made by generations of Hispanic Americans to our country. So this could be scientific contributions, cultural contributions, social justice contributions, business contributions, artistic contributions, athletic contributions. And as we move through our biology course this month until October 15th, I will be bringing up some of this history, especially as it relates to biology. And in fact, one of the most important biologists, not only in the history of the United States, but also in the history of the world, is of Hispanic descent. And he actually was the first person to theorize about climate change. And he is a Mexican American. So we'll talk about him and his story and his scientific findings and why they were so important and why it actually won him a Nobel Prize. But this is a holiday that we celebrate across the country, and we have celebrated it for the last 32 years. So not nearly as long as you might expect, only since 1988. And that kind of speaks to some of the history of our country, which is a history that has been tainted by racism and xenophobia, especially xenophobia aimed at people of Hispanic origins as they are entering our country.
What is xenophobia? I never heard of that. Sorry.
Excellent question. You know, I love questions. Some of the best minds ask the most questions. Xenophobia is fear of foreign things. So we know that phobia means fear. Xenophobia is fear of things that are foreign. So in this case, in most cases, you'll hear the phrase xenophobia to refer to fear of foreign people, people who are not from or who are not native to that country. The irony of it is that, you know, in some cases, Mexican American people were native to the United States. You know, Texas and New Mexico and Arizona and California were Mexican territories, you know, for the actually, you know, for the majority of history. And so there are people who are, you know, of Mexican descent who were here before the Europeans were here, but still we consider them foreign. So it is a topic that deserves us talking about it and learning about it because, you know, we should be celebrating the contributions while also acknowledging the fact that our country has, you know, through policy, behavior, and actions been very harmful to some of those Hispanic communities over the years and continues to be. So it's something that we'll talk about, and I'm going to try to touch on multiple sides, and hopefully I get an opportunity to learn from you all and your experiences and what your perspectives are. But this is something that I'm excited about. And the reason why it starts on September 15th instead of September 1st or October 1st is because today is actually the anniversary of independence for 5 Central American countries: Costa Rica, El Salvador, Guatemala, Honduras, and Nicaragua. And if you know anything about geography, those countries are all very closely located on the map, and they're all in Central America, and they all celebrate their independence from Spain on September 15th. Tomorrow is Mexico's celebration of its independence. September 16th is not actually when Mexico won its independence from Spain, but it is when it began its fight, its struggle for independence. So I appreciate the fact that September 16th is the day that Mexico celebrates that beginning of the struggle instead of just the end of the struggle, because to me, the struggle is what is most important. And perhaps you've heard of La Raza or La Revolucion. And these are phrases that really, to me, speak to that struggle. And I also am aware that Chile is celebrating its independence on, that'll be Friday, September 18th. Friday. So September, especially the latter half of September, and then we've also got a couple of holidays in early October that deserve recognition. That's what makes this time an important time in Hispanic heritage. And some of you might be wondering, what's the difference between the phrases, the terms Latino and Hispanic? And one thing that I want you all to be comfortable with is asking people about their preferred identities and how they would prefer you refer to them. So I think it's a good idea for us to be comfortable saying, hey, you know, how would you prefer that I refer to you? How would you like me to refer to you? Some people prefer Latino, some people prefer Hispanic, but there are some small differences between them. For example, Latino is a phrase that talks specifically about people who are from Central and South America. So it does not include people from Spain. And then on the other hand, Hispanic is a word that describes people who are Spanish speaking or who are descended from Spanish speaking people. So it includes Spain, but it does not include Brazil, where the majority of people are descended from Portuguese speaking people and they speak Portuguese. And it does not include French Guiana, which is a country that had been colonized by the French, obviously, and so they are descended from French-speaking people. Same thing with Haiti and Martinique and Saint Martin. They are French-speaking people, so we don't consider them Hispanic, but they are still Latino. And of course, you can be any race and still be Hispanic, and still be Latino. There are people in Brazil who are white. There are people in Brazil who are black. There are people in Mexico who are white, and there are people in Mexico who are black, and there are people in French Guiana who are Asian. And so you can be any number of races and be from any one of these countries. But I think it's important that we know the difference between these terms and how it applies to some folks and how it doesn't apply to others, but also that we've got a lot of people who really it's a matter of choice. And so you should be comfortable asking them, what is your preference? How can I make you feel most comfortable? All right, so that's what I wanted to talk about today as it relates to Hispanic Heritage Month, but it is not going to be the end of the conversation, and we are going to relate it specifically to biology. So I'm excited about it. Last thing, Latin X is often used and increasingly popular because it is a more gender inclusive term. Some people actually prefer to be referred to as Latino or as Latina, but Latinx is a phrase that has now been created in order to represent people who do not fall into that binary of gender, who are not who wouldn't consider themselves masculine and wouldn't consider themselves feminine, and so therefore they want that Latinx phrase because it doesn't have the O or A ending to imply gender. All right. This is kind of, well, I'll come. Do I want to show this right now? No. I'll come back to that if we have time. Okay. So just as review, sorry, the first phase, and that should be phase 1. The first phase of the cell cycle, of course, is interphase. Interphase is made up of 3 parts. And Ayana told us we've got G1 or gap 1. That's a period of cell growth. Then we've got synthesis, which is a period in which the DNA is replicated. Then we've got G2, which is gap 2. There's another period of cell growth as the cell prepares itself for the second phase, which I believe Castro told us as well as Alicia, is M phase. And M phase can be mitosis or meiosis. Am I recording? Yes, good. And then phase 3 is cytokinesis, when the cell moves apart. Now, one thing that we didn't mention yesterday, but it's actually in this image here that I'll talk about very quickly. There's a G0 or G0 phase that we don't talk about too much in biology, but you all are an honors class. So I think this could interest you. And if you're considering taking the AP class next year, we do talk about it in that class. But G0 is a phase where actually a lot of cells spend a lot of their lifetime because it's a phase where they're not really doing much. They're just kind of carrying on with their daily functions. They're just making a lot of proteins. They're not trying to divide. They're not trying to reproduce. They're not copying their DNA. All they're doing is just making proteins and then sending them out. I apologize if my background is getting loud.
Mr. Rudd?
Yes.
Rudd? Can you explain a little bit more about the checkpoints again?
Yes, good question. So the cell's DNA tells it exactly when to ask specific questions of itself. And those questions come at different points in the cell cycle. And the whole point of it is to prevent the cell from making errors in the cell cycle. So, for example, in G1, it's called a restriction point because you first, before you copy the DNA during synthesis, before you replicate the DNA during synthesis, you want to ask yourself, am I okay to do that? Is my environment healthy? is the DNA intact? Because if you copy DNA that is not intact, or if you copy DNA that has some mutations in it, then you not only jeopardize that cell, but you jeopardize other cells. And this is how we end up with cancer as well. So we've got to ask ourselves these questions. And then later on in G2, we might want to ask ourselves, is the DNA correctly replicated? So after synthesis, we need to know, did synthesis happen correctly? If it hasn't been correctly replicated, well, then we don't want to reproduce another cell with that erroneous DNA. And then lastly, before mitosis, are the chromosomes ready for separation? So are we ready to actually split into two new cells? If not, and this does unfortunately happen in some pretty detrimental ways that can lead to a number of conditions like Down syndrome, we can end up with a cell that gets split and has an uneven number of chromosomes. So instead of having, just to make the numbers simple, instead of having 2 in this one and 2 in this one, we could end up with 3 in this one and 1 chromosome over here. So this one has 1 extra chromosome. This one has 1 less chromosome than it should. So we need to make sure that the chromosomes are aligned in the correct way and that they're ready to be pulled apart. If not, we can end up with some pretty bad situations.
Thank you for explaining that, and I'm sorry again for taking your time.
Oh, no, you're fine. Good question. I think other people probably had that same question. So I am proud of you for asking it. Let's see, where was I? All right. So nothing to write down on this slide. But we talked a little bit yesterday about the difference between sexual and asexual reproduction. But what do you all think are some additional differences between the two? What are you aware of?
The asexual reproduction, they don't have gender.
Okay. Yeah. Sorry, did you have more to say? I thought I cut you off.
No.
Okay. Yeah, so
I'm
we
still
don't.
confused on how asexual reproduction is. Like, how does it work? Like, I don't know how it works.
Okay. We will talk more about it. Asexual reproduction, kind of like what Alicia was saying, we don't rely on 2 mates. So we don't need a mate that has sperm and a mate that has eggs. We just need, it only needs 1 individual. So this is how most bacteria, most unicellular organisms are able to reproduce asexually. So that means that they can literally just make copies of themselves. Sexual reproduction, on the other hand, does require 2 individuals, 2 different organisms. 1 organism needs to be the source of sperm, and 1 organism needs to be the source of an egg. Even with plants, this is the case. So pollen, even if you can think about in the early spring months when you see a lot of pollen floating around in the air, that is actually essentially plant sperm that is hoping to germinate another plant or fertilize another plant. So sexual reproduction requires 2 organisms. Asexual reproduction really just requires 1. And we'll talk more about them. Okay, here's a quick joke. What did the cell say when his sister stepped on his foot? Mitosis. I know, it's not great. And the fact that you don't get it, mitosis, like MYTOE.
That's not a very good joke.
Stephanie thought it was funny. Anyway, yeah, I know. It's a tough crowd today. All right. So here's what I want you guys to write down. So keep in mind, mitosis is the M phase, which means it happens second. It happens after interphase. We need to replicate the DNA before we can undergo mitosis. We need to copy the DNA before we can undergo mitosis. But mitosis is the cell division process that makes somatic cells. And if we remember from yesterday, somatic cells are body cells, like muscle cells and nervous cells and stomach cells, skeletal cells. So mitosis makes somatic cells. And we can see this process happening in a very basic animation right here. As one cell is beginning to split, we can see that from one nucleus, they're sharing a nuclear membrane, but it's starting to split in half, and eventually we end up with two daughter cells. So we've got one parent cell that, through mitosis, produces two daughter cells. Okay. So mitosis is the process that is used for that asexual reproduction. And it allows cells to make identical copies of themselves. They have the same DNA, they do the same function, they are shaped the same. They are literally like if you put in a paper in a copier and out comes an exact version of that copy, of that paper. Okay. So mitosis undergoes one round of cell division to make two identical diploid daughter cells. I know that's a lot, but we can break it down. So we already talked about the fact that one parent cell makes two daughter cells. We already talked about the fact that those two daughter cells are identical. They're exactly the same. They're exact copies of each other. But this word diploid is new to us. A diploid is a cell that has pairs of chromosomes. And so often you'll see us refer to this notation here, 2N, to represent a diploid, because it has twice the number of chromosomes. It's got one from each parent. keep in mind, we're talking about somatic cells here. If you remember from yesterday, we also introduced gametes, which are sex cells. But in this case, we're only talking about the somatic cells. And we can see this is an important diagram down here. We start off with one mother cell, one parent cell. First, the DNA is replicated. So we copy our DNA. Then we undergo a process of cell division. This is mitosis right here. And then cytokinesis allows the cells to split apart. So we've got interphase, where the DNA is replicated. We've got mitosis or M phase. And then we've got cytokinesis. Okay. So just like in interphase, we had those three steps. We had G1, S, and G2. Mitosis can also be broken down into 4 steps. That's Miss Fields' Bitmoji character. There is prophase first, metaphase, anaphase, and telophase. Telophase is last. Telo again means end. So end phase. Pro means before or beginning. So prophase is the first step. I know this isn't highlighted, but I definitely want you all to write this down, please. And as you all think about, good study skills, good study habits, or just habits of being a good student in general, one thing you maybe have used before is creating acronyms for yourselves. And you can use this PMAT to create some useful acronym. My first year here, my students came up with the acronym for IPMATC. I is interphase, then prophase, metaphase, anaphase, telophase, and then cytokinesis. And they came up with this acronym, I play music at the club. But you can come up with your own if there's something else that you can think of that's helpful to you that will help you remember the steps. That's really one of the good skills, stronger skills of a strong student. Okay. So this first step is prophase. Prophase is an important step. Oh, hold on. So prophase is important because the nuclear membrane needs to dissolve. That means that essentially the outer layer of the nucleus dissolves, and this allows the chromosomes to be accessed from outside of the nucleus. We also have what are called centrioles and spindle fibers. And this is not, and I know there are a lot of new terms being thrown at you right now. Really, the most important part of prophase is that the nuclear membrane dissolves. But these centrioles and spindle fibers are essentially kind of like the motors that eventually are going to pull the chromosomes apart. Are there any questions up until this point? Anybody have any questions?
No.
Okay, thank you. Okay. So during metaphase, meta means middle, the chromosomes literally line up in the middle of the cell. And the spindle fibers will attach to their centromeres. They'll attach to the very center of the chromosomes. Okay. During anaphase, ana means apart. So those spindle fibers, which have been attached to, they're attached to the chromosomes on one end, and they're attached to the centrioles on the other. The centrioles basically reel them back in. The centrioles reel the spindle fibers back in. And in doing so, they're pulling the chromosomes apart. So what used to be two sides of an X get pulled apart. And now you just have sister chromatids that are floating away from each other. So those centrioles are just like the reel of a fishing rod. They pull the spindle fibers in, they reel the spindle fibers in, and by doing so, they pull the chromosomes apart. And then lastly, we've got telophase. And this is the step that comes just before cytokinesis. So this is as the new daughter cells begin to form and two separate nuclear membranes appear. So right after the M phase, we've got cytokinesis. So right after the cleavage of telophase, we've got cytokinesis as the two new daughter cells split and move apart from one another. Okay. So here it is. Here's a good image. And as we think about being efficient and effective note takers, it may be a good idea for you to try to recreate this image or some similar image in your notes where you draw a circle like a pie chart, and you can represent different sections of the pie chart as different sections of the cell cycle. You start with interphase. And then you move into M phase or mitosis, which has 4 steps: prophase, metaphase, anaphase, and telophase. And then you end up with cytokinesis. Okay, so looking at these images and using the notes that you all have, what would be the correct order of cell division? What do we think is?
Mr. Rudd, I was
Yeah.
still copying down the pie. I mean, not the pie. Okay, I got it down.
Okay, so if you look at these images and compare to what we just talked about in your notes, what should be the order of them? I know we only have 4 steps of mitosis, but I'll give you a hint. We really have 2 telophases in this image.
Is 2 first?
2 should be first, yep. So 2 would be prophase. I guess we have 2 steps of prophase, not 2 telophases.
Then 4?
Yep, then 4. So we can see in step 2, let me see if I can get my, in step 2, we can actually start to see some of the nuclear membrane is breaking down. Then in step 4, we see the spindle fibers starting to align. Then what happens?
3?
Then we've got step 3. This would be metaphase. As you can see, the chromosomes are lined up in the middle of the cell.
1?
Then 1, this would be anaphase, as the chromosomes get pulled apart by the centrioles. And then what?
5?
Yep, and that leaves 5. So this would be telophase. As 2 new cellular membranes form, a nuclear membrane is reforming around the chromosomes, and you're ending up with 2 new cells. All right. So for humans, our somatic cells have 46 chromosomes. We've got 23 from each of our parents. So once our cells undergo DNA replication, we've got 92 chromosomes. We've got twice the number because they've copied themselves. But then when they undergo mitosis and they split apart, we end up with that original and correct 46 again. So this is what you want to see happening. You've got to first double it before you split it in half again. So we end up with the exact same number of chromosomes in the daughter cells as we did in the parent cells. Okay. So with a banana, what would need to happen? What would go in this? How many chromosomes would this cell have after it's been duplicated?
28.
28. I thought it was going to appear here. 28, and then once it splits again, how many would these new daughter cells have?
14.
14, good. Okay, I'm skipping this. All right, so that's it for the synchronous portion. We have 20 minutes. You have an exit ticket and a mitosis practice assignment. Both of them are very short, so you should have plenty of time to go ahead and knock them out. Please let me know if you have any questions or you run into any problems while doing that. I did just recently check them. So from what I can tell, they are good to go. Let me know if any of the images, there's only one image. Let me know if it doesn't load. Okay, folks, it is 11:35, so that means that it is the end of second block. Please let me know if you have any questions between now and tomorrow. But other than that, have a good Tuesday.
See you tomorrow.
See ya.